Hello, everyone. I am Kenny Coogan. I'm the Education Director for the International Carnivorous Plant Society. And thank you, everyone, for joining us. Even Monica is joining us, Maksym. And I'm excited to have Monica and Maksym, and maybe some other Slovakians joining us for this month's webinar. If you are attending this live, please feel free to ask questions in the chat below. And if you're ever in Slovakia, ask Maksym and his wife Monica for a tour. They are wonderful, wonderful tour guides. Dr. Kadim Gilbert and we visited them after the conference, and it was so great. And Maksym, I'm going to let you introduce yourself and tell the people about your credentials. And I am looking forward to your presentation.
Thank you, Kenny, for kind introduction and also for inviting me to speak in front of this wonderful audience for International Carnivorous Plant Society. I'm really honored to present, to be here tonight and present a story of the secret passion already from my college years. I'm Maksym Danchenko, and I'm currently leading an emerging group at Nitra, Slovakia. And here you can enjoy the view and see the view on the city from the hill with just above it. Slovakia is a tiny country, however, it has astonishing natural beauty. There are high mountains with rocky peaks, and there are different seasons, including winter, so you can enjoy both snow and heat. There are caves, streams, lakes, a lot of nice things you can experience in Slovakia. Currently, I'm working at Nitra, which is a regional center with a rich history, and I would call it vibrant academic life. There are 2 universities and several state research laboratories. I would like to say a few words about my past career. I did a degree in plant physiology in Lviv, Ukraine, working on woody plants and possibilities of using them for remediation. After that, I moved to Kyiv, capital of Ukraine. There I did a PhD in radiobiology, looking how agricultural plants survive in Chernobyl. After that, I eventually moved to Slovakia. There I dived into sea of proteomics, working first at the Institute of Virology. Then there was short experience at biotech company Axon Neuroscience, where we contributed to looking for biomarkers of Alzheimer's disease. And currently, in parallel to working at Nitra, I'm also having a role at Bratislava at Institute of Chemistry. There I'm helping to manage advanced analytical infrastructure, including high-performance liquid chromatographers and mass spectrometers, which are indispensable for modern proteomics. Meanwhile, I also enjoyed short yet really insightful internships, both to neighboring European countries, but also overseas. In my lab, currently we are working on several topics. One of them is resistance of aquatic plants to ionizing radiation, and in parallel to pests and pathogens. We have a hypothesis which we were able to confirm also experimentally that because of chronic ionizing radiation, plants are getting more susceptible to pests. And I hope that pretty soon we will be publishing an experimental paper showing all of those mechanisms. I'm collaborating with the colleagues from the Institute of Hydrobiology on this topic. Another story which we develop is more practical one. It deals with wheat, important staple food worldwide, and how is it adapting to drought, but also recovering from it. Here we are employing proteomic approach based on redox proteome, which is pretty common, and post-translational modification, which we believe is critical for regulating the processes during growth. Again, I have very strong collaborators, expert physiologists for this project from Institute of Plant Genetics and Physiology. Next to the last story is about another unique group of plants, parasites, and we are trying to understand what is exactly happening when they germinate. This group of plants has unique germination procedure. They need to perceive stimulants coming from potential host plants in order to trigger their germination. So we want to explore the mechanism. So it's still very much, this project is still very much in the infancy, using phosphoproteomics, and then confirm functional relevance of the changes using transgenesis and regeneration. Well, this sounds exciting. Nonetheless, it's pretty challenging because the parasite is not at all easy to germinate. And in the past, my colleague just succeeded this once in 100 of attempts. For all of these stories, we are using our core technology to use is proteomics. We rely on fast yet efficient sample preparation using magnetic beads and detergent-containing buffer. Then we profile the digested peptides on chromatographer, Vanquish Neo, using reverse phase columns and long gradients. The data is acquired on up-to-date mass spectrometer Exploris 240 with so-called data-dependent acquisition. And we rely on community developed and supported software packages such as MaxQuant, Perseus, MapMan. And all of this infrastructure is available at the Institute of Chemistry. There I'm also partially employed. At our institute, we are having, let's call it a small collection of Droseras, and I will show you some pictures of them. First on the stage is Drosera indica. We are growing plants in vitro on solid medium. And yeah, and here you can enjoy those pretty plants. Another species to display is round-leaved Sundew. This plant it can be also naturally seen in Slovakia in swamps areas. We are actually planning a field trip in a couple of weeks to see those plants in natural habitats. Back to the essence of the talk, and my major focus today will be on development of leaves and digestion of prey in carnivorous plants, taking as an example Sundews. This is very large underwaist genus. Plants are mostly grasses, and they can be distinguished rather easily just looking on them, because the morphology of their leaves is usually, yeah, well, pretty distinctive. And Sundews are having flypaper type of traps. So first I will say a little bit of background, then explain objectives of our project, the methods we are using. I will tell our first results and also draw future prospects for the study. I was recently I came up to the paper from international collaborative group of experts in the field, and the colleagues are suggesting to look on trap leaves as analogs of the mammalian digestive system. However, the difference here is that while in mammals and other animals, the digestive system is divided into mouth part, or trapping, so to say, and chewing, then stomach for releasing acid, digestive enzymes, and intestine, which is absorbing the nutrients. For carnivorous plants, trap leaves are all-in-one organs, which are capable of performing all of these processes. Of course, for mammalian systems, there is much more information about how the digestion works. Much less so is known in about about carnivorous plants. You can see multiple question marks here. Yet it is proven that digestive fluid is acidified in the same way as in mammalian cells. Enzymes are secreted, really different groups of enzymes, including proteases, phosphatases, esterases, chitinases. So literally everything what is needed to decompose the prey. And then it is also known that the substances can be absorbed both through specialized transporters, but also through endocytosis. So here comes the main rationale, hypothesis, and objective of our study. We believe that using multiomics, we will be able to pinpoint mechanisms of development of trap leaves and also get insights in how exactly the digestion is happening. So for that, we first need to select different developmental stage through like primarily looking for morphological differences. Then we are going to do combined multiomics, including proteomics and transcriptomics. And we are doing two parallel experiments. One is about leaf development, another one is about induced digestion. The problem with digestion is that it's also not very known, the time frame is not very well known. Is it happening within a day, 2 days, a week, or probably just a few hours? This is another goal for us to elucidate. In order to put this mechanism into plant physiology perspective, we will look for phytohormones, and our ultimate ambition is to characterize differently accumulated proteins or genes using first in silico approaches, but after that also experimental approaches with cloning, purifying, and characterizing those proteins. We decided to use a variety of different methods, starting from cultivation, followed by proteomics, transcriptomics, phytohormone profiling, enzymatic assays, and analysis relating to production and purification of recombinant proteins. In our lab, we cultivate plants in aseptic conditions. So basically all manipulations are done in the laminar flow hoods. Since we are using media with nutrients, including also sucrose. So to avoid the contamination, we have to do our experiments in a laminar flow hood. After that, we grow plants in cultivation rooms where we use photosynthetically active blue and red radiation. So basically, we are using 25% Murashigin medium, which is relatively more pure in for nutrients than, let's say, normal one, because carnivorous plants are less demandable for nutrients for their growth. So in our study, we focused on forked Sundew. And while cultivating it in vitro, it is possible to observe kind of morphologically distinct stages. Firstly, the plants are proliferating widely, forming forming lather small clamps. Then we are entering vegetative phase. There we look, let's say, much more normal, more like in nature. And finally, if you keep them cultivating for longer without dividing into new plates, into new boxes, we start even to flower. For a digestion induction study, we use two approaches. Firstly, we used combination of two polymers, chitin, which is component of natural prey for Sundew, like insects. We have exoskeleton, which is built of chitin, but also pachyman, which is a little bit strange because let's say plants are not really coming in direct contact with this polymer from their prey. However, curiously, the data from my colleagues showed that Pahiman is a particularly strong inducer of digestive enzyme. And also we try to use more natural inductor, which is larvae, actually ground larvae in not intact larvae of the mealworm. Just to take a little bit of a break from the science, I'm showing you another beauty in our collection, which is Drosera Cape Sundew, Drosera capensis. Next one is coming pretty robust, spoon-leaved sundew. We often even can observe its flowering in culture condition. And finally is forked sundew, the one my main research is focused. Okay, so back to the science. For the leaf development study, we closely observed what is going on, and we were able to select one, let's say, a little bit strange stage which we called base or basal. And this sort of heart-shaped leaves are different from normal adult leaves of Drosera binata, and we are normally observing them closer to the base of the plant. And after that, we selected, let's say, 4 different developmental stages of normal leaves. First one is so-called roll leaf, where you basically see just a stalk, which is still rolled. Then something like heart shape, we call it stem leaf, is coming. When the fork is starting to appear and already you can see tentacles, however the dewdrops on the tentacles are still translucent, we are dealing with fork leaf. And finally, fully developed mature leaf we call tentacle. It's completely open, unrolled, and the tentacles and fluid on them is becoming purple. Actually, forked sundew is a good object for our study because we can really very nicely distinguish those several morphologically distinct stages that would be far from straightforward for other sundew species. Okay, so we are going into our findings. So using proteomic profiling, we were able to identify more than 1,000 proteins, and among them, more than 400 were differently abundant between different developmental stages. Obviously, if you do biology, you need to do biological replicates, and we did that. And those circles you can observe are actually encircling different biological replicates of the developmental stages of leaf. And you can see that the reproducibility of our analysis was like super good. From this so-called principal component analysis graph, beside reproducibility, we can also assess how different different sample groups are one from another. And we can spot that those basal leaves are more similar to mature tentacle leaves, but they're still forming pretty distinct and separate cluster. Other than that, we had very nice line from early development, like roll stage, into stem stage, and then fork stage. We are just in the beginning of the data analysis, and first of all, we look for the cluster of proteins which were constantly accumulating during the development, starting from literally not existent and going up and up and up, and with the highest quantity on tentacle stage. Here is a list of them. We are right now thinking about what can be the meaning and how exactly we can contribute to biology. For example, developmental occlusion protein B is the one which is regulating transport through plasmodesmata and basically loading of the phloem. We believe that during the development, the flow of assimilates is getting lower. So probably direct photosynthetic function of leaves is getting smaller. Then dual specific kinase is a protein involved in phosphorylation, which is a very well known signal mechanism. Then there is a lot of data in literature which is binding RAS-related proteins to different developmental and growth processes. Then I will show you one specific hypothesis which we tend to explore a little bit more in deep, and it's related to lipoxygenase. And for example, one study showed that Arabidopsis overexpressing lipoxygenase from cucumber was able to attract wasps if the plants were attacked by pests like larvae of white butterflies. Well, here is the data which is clearly showing preference of infested Arabidopsis, let much more wasps were attracted in this case. And we believe that something similar can work in carnivorous plants, that most likely one of the plausible explanations of accumulation of lipoxygenase during the development is actually creating secondary metabolites which are plausibly attracting the prey. But obviously this is the subject for further functional studies. In a trap induction study, we observed some curious phenomena. So over here, I'm showing you just control plants and plants which were induced by the mixture of polymers. It was actually working real well for us. You probably remember that as an alternative inductor, we used bugs. But with bugs, it was not that well. Funny enough, upon analysis, most of the proteins which we were able to detect were actually coming from larvae. What was super strange, initially I had, not that much of explanation about that. However, now I'm a little bit playing with the idea that there was a massive endocytosis, but this is something what definitely needs to be confirmed and explored. Anyway, results with the larvae was strange, so we are thinking about how to redesign the study. But with polymers, we saw again very nice reproducibility among the samples, and we also saw like real big change upon applying inducers. However, another curious finding was that we used actually inducer like short treatment 1 day and 2 days. And as you see, these groups are super close one to another. So meaning that 1 day was enough, literally nothing additional happened during another 24 hours. So we want to go now in two directions, both into like looking earlier, but also looking later, like probably checking if digestion is already starting after 6 hours, or let's say what is happening after 4 days. In this data set, in this rich data set, we were primarily looking for hydrolysis. Because in our department, there are other colleagues who are extremely keen to see candidates in order to work on them for cloning, purification, characterization, and potential using biotechnology. And in fact, we were able to spot quite a number of enzymes. Some of them are like really skyrocketed, others not so much. There was one actually curious finding. We haven't seen any induction with some enzymes which other researchers reported to be induced on mRNA level. And here, like for example, hitinases. And here can be a few explanations, like one thing is that something what is expressed to mRNA is not necessarily translated to proteins. And another explanation, which I'm thinking is much more plausible, is that perhaps mature leaves are already containing enzymes which are simply not active unless the fly appears in the trap. So they are probably activated, like one of the plausible mechanisms is actually acidification of digestive fluid. To complement this proteomic data, we started to work on enzyme activities, starting from glucanase. And one take-home message for us was that if you have a protocol published in the literature, super sensitive, super easy, not necessary it will be so in your situation. For example, when we tried to adopt this dinitrosalicylic acid-based protocol, for us, the sensitivity was 2 orders lower than published in the literature. So I'm not really sure how to explain it, whether the quality of reagents we are getting here is not sufficient, or probably not, or probably not everything what is published can be 100% reliable. We are still in the stage that some experiments are not accomplished. Like next step for us is doing transcriptomics. We have optimized extraction protocol developed in our institute. We negotiated with one reliable company, the sequencing service, and we are still applying for funds in order to get this experiment done. We agreed with a collaborator from Olomouc, from Czech Republic, who's gonna help us with phytogramon analysis using high performance liquid chromatography. And we also designed a strategy how to isolate genes of interest, clone them, and express in bacterial, but also in eukaryotic expression system. So to sum up, Here today, I told you that the career path in science can be bumpy, and finally, I arrived to studying carnivorous plants after more than, probably more than 20 years just dreaming about that. Nitra and Slovakia and Plant Science and Biodiversity Center are amazing places to live and work. We have at the moment rather small but growing collection of Sundews. For leaf development, we clearly show reproducible and dynamic proteome profiles. And of course, we were able to identify proteins related to classic functions like photosynthesis, defense related, which are often repurposed in carnivorous plants for digestive purposes, also a number of enzymes. And we are looking how to interpret all of this data. Interestingly, we showed that during the induction, 1 and 2 days are literally the same for forked Sundew. And now we are like really looking which hydrolytic enzyme is promising to further characterize. I'm looking for any sort of feedback to my talk and sharing ideas for collaboration. I want to thank my existing collaborators, Dr. Gutko, who is helping with ionizing radiation, Dr. Stasik, excellent physiologist, instrumental for drop-related study, Dr. Matishova, really well-renowned researcher in parasitic weeds, who in the past did a lot of bright discoveries, Dr. Barat, proteomics guru, Dr. Libantova, who have been working with Sundews and their biotechnological applications for a long while, and Dr. Danchenko, who happens to be also my life for helping with making this presentation much more visually appealing. The research in the lab is supported mostly by local funding agencies like IPUV, Vega, Dr. Grant, VIA, but also European network cost. If having any suggestion, questions whatsoever, feel free to reach me. And here is my group. Actually, people who are working on carnivores are Oля and Rohan. Others, like Marina, is working with parasites, Shubi with ionizing radiation. Ahmed was a visiting professor who just left. He was working with, well, he's still working with plant viruses, and that was also a recent topic in the group, and he will be substituted by new student Umar. Thank you, and looking forward for discussion.
All right, so Maksym just did a wonderful presentation. We like to do at least one a month. And the way that I met Maksym was at the 2024 ICPS conference. And right now we are looking for a host for the 2026 ICPS conference. And it's going to be in the Americas. It's going to be in North America, Central America, or South America. If you have an organization or a university that you think can host, please contact Brent, who is our conference chair. He's our conference chair. His email is brent@carnivorousplants.org. So Brent is on the board, and he manages all of the conference applications. And then we go over to see who is best equipped to handle a conference. I know I posted this a couple of months ago, and we got lots of feedback. Everybody saying they would love to have it in Central or South America because we've never had it there, and we've had several in North America. But the thing is, we need a host. So yes, we agree that we would love to go to those regions, but we need somebody to help us host the conference. So if you are that person, please contact Brent. The ICPS is a group of professionals like Maksym and also backyard growers and scientists from all around the world. We have over 1,100 paid members. We host monthly webinars. And if you're interested in presenting for the ICPS, you can send me an email at kenny@carnivorousplants.org. You could be somebody who grows carnivorous plants in their bedroom, or you could be a scientist who is trekking the mountains of the Philippines and discovering new species. We're looking for all types of experience and expertise. If you are a teacher or if you're getting into the hobby, we have 5 animated videos that we encourage you to check out, which teach you how to grow carnivorous plants indoors, outdoors, how to feed Venus flytraps by hand, things like that. And we also have worksheets that accompany those digital animations. If you want to become a paid member, you can go to our website and you can see our different membership levels. When you become a member, one of the benefits is you get a beautiful full-colored newsletter 4 times a year. To check that out, you can go to the website below. And another benefit of becoming a ICPS member is our seed bank. We can usually ship internationally. Once you become a member, you can sign on to the membership website and you can see the regulations for that. And if you are somebody who is breeding carnivorous plants and you have surplus seeds, we encourage you to donate your seeds to our seed bank because our seed bank is only as good as what our members make it. So this is all done through donation. And it can be a really good fundraiser for our organization. And then what we do with the proceeds, because we are not for profit, is we can do cool things like Carnivores in the classroom, where we fund over 50 teachers every year to add carnivorous plants to their K through 12 classrooms. And we've been doing that for a couple of years now. I think we're on the 4th or 5th year. And you can see the hundreds of students that we've been able to impact. For this year, these are the participating individuals and nurseries. We can fund over 30 teachers in the United States, and we can fund a couple of teachers in Europe and Australia. We are doing the Carnivores in the Classroom grant every year in the month of August 1st through August 31st. If you are a nursery or an individual who can fund classrooms, please send me an email at education@carnivorousplants.org so we can provide even more carnivorous plants to more schools, because you can probably imagine when you first got that spark of interest, when you first learned about carnivorous plants. And that's what we want to continue to do. We want to inspire young people to care about botany, specifically carnivorous plants. And so we like to fund education initiatives, but we also like to do conservation initiatives. If you're getting our newsletters or if you're following us on social media, you'll see that we post the different governmental and non-governmental organizations, NGOs, about how we support carnivorous plant conservation in situ. And one way that you can do that is by you can just donate money to us. Well, what's even more fun is you can go to our online store, which is a drop ship service, which means it creates these things all around the world and ships them to you. And you can get cool merchandise with butterworts, cephalotus eating the world, my favorite Bicalcarata. We have Utricularia, Helianthera, Drosera. And you can get mugs and totes and t-shirts and bags, and all of that also supports our conservation and our education initiatives. And with that, thank you very much. Okay, so Maksym, Maureen says, is it is there a YouTube channel for ICPS? And Maksym, can you see me or am I invisible right
I see just a white background.
now? How exciting.
Still the same.
Yeah, you can see
Oh,
me.
okay. Now you're here.
That was fun. So Maureen wants to know if we have a YouTube channel. Yes, if you just go to YouTube and then type in International Carnivorous Plant Society, or if you go to youtube.com/ICPSTV, then you can see maybe hundreds of videos of past webinars, World's Carnivorous Plant Day things, our animated videos. Yes, Maureen, we have lots of YouTube. We have lots of things on YouTube. Okay. Will Lush says, thank you for the presentation. And then Louis Williamson says, hi there, Maksym. Great talk. I previously worked on population phylogenomics for Drosera binata across Australia and New Zealand. There is at least one deep molecular divergence within the species. Do you know where your plants are sourced from? It'd be interesting to know if there are differences between these major population clusters. Go ahead.
I'm not 100% sure, but I do believe that we have Australian ones. At the moment, I'm not really in the area of looking for these differences between the populations. It's not yet at the moment a focus of my research, at least of Sundews. Maybe it will be at one point. However, for example, what we are also trying to explore is the difference between in vitro grown Sundews and in vivo. So like in pots, so let's say in the nature, in the garden. And we had some naive observations, like for us, it seems that those in vitro ones are much more lazy. So they don't want to eat that much. So when you give them prey, their movements are slower, and some species which are capable to move are not really moving. So this is one interesting thing. And then another thing which we will probably explore, like one colleague of mine wants to look into differences between ecologically different Sundew species. The one which are growing into desert comparing it to tropical rainforest one, comparing to classical swamp ones, in order to kind of see what are the digestion mechanism differences among them.
All right. Thank you so much, Maksym. And I just posted our YouTube channel in the comments. Last month, we had the opening presentation from the ICPS conference. We had them do a webinar for us. And then next month, we're going to have Dr. Yana Lebentova, which is Maksym's coworker.
Indeed.
Also do a presentation. So we are excited for her. And thank you so much, Maksym. And I hope lots of people, once we publish this on YouTube, reach out because, you know, as you were saying, science is bumpy. And I really like how your talk really highlighted the fact that evolution just repurposes and not reinvents. So if a plant already has an enzyme or a protein used for something, they can repurpose it to maybe lure and attract their prey.
Indeed.
All right. Thank you so much, Maksym. And thank you, everyone who attended this live. And I hope to see everyone next month.
Yeah. Thank you, everyone.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.